Welcome back to another video tutorial from Aditya Biologics. So guys, today the topic is apoptotic pathways. Okay, and this will be the second part of videos on apoptosis here. In the first part of the video, we have discussed about the apoptosis, the basic concepts on apoptosis, why apoptosis is important, yeah, and how it takes place, what are the processes. We have already discussed the topic, yeah. And today in this video, we are going to discuss the pathway, yeah, the major pathways of apoptosis, yeah, and it is one of the very important topic to discuss here. So stay tuned with the video, and yeah, make sure to watch the video on apoptosis, the part 1, yeah, and the links are given in the description box. Okay, so now let's start the video. Okay, so first going through the video, let's start with the basic concept on apoptosis, yeah guys. Okay, so here you can see apoptosis is a pathway of cell death that is induced by a tightly regulated suicide program in which cells destined to die activate enzymes that degrade the cell's own nuclear and cytoplasm proteins. Yeah guys, so in simple way if I say, then apoptosis is nothing but a programmed cell death in which cell intentionally takes a decision of committing suicide, yeah, whenever there is any abnormality in the cell, yeah, or any stress going on inside the cell. So for the betterment of other neighboring cell and as a body as a whole, the cell decides to commit suicide, yeah, the cell dies itself on its own. It's a natural process. Okay, and the overall concept we have already discussed. Okay, now let's move on. Okay, so these are the basic pathways you can see, yeah. This is the apoptotic cell. It's the here nuclear fragmentation is started, and here you can see the blips. The blips are being forming, yeah. These are the blips. The bubble like or buddings are formed, yeah, bubble like structure. The same process continues and the cell disintegrates, yeah, and these cellular debris, disintegrated cells, are engulfed by any of the phagocytic cells, and mainly it is macrophages. Okay, macrophages. So this is the basic idea. Okay, now let's see more. Okay, now let's start with the mechanism of apoptosis. Okay guys. Okay, so the mechanism of apoptosis is divided into two major phases, yeah, and this mechanism is very important, guys. You need to understand very deeply, so pay attention. Okay, so the phases are the initiation phase and the execution phase. Okay, initiation phase, then the term itself says initiation means here the initiation of apoptosis takes place, means all the preparation for the apoptosis like activation of proteins, factors takes place in the initiation phase. Okay, and in the execution phase the real cell death takes place. Okay, and we are going to discuss these both phases one by one. Okay, so now let's start. These two different phases initiated by distinct stimuli, and they involve different sets of protein, although there is some crosstalk between them, yeah, and we will also discuss about the crosstalk between the phases. Okay, now both phases eventually converge with the activation of caspase, which are the actual mediators of death. Actually the caspase talking of here is caspase 3, guys, the very important death protein. It is actually caspase 3, and it is only responsible for the disintegration of cell and apoptosis process actually, yeah. Okay, now let's move on. Okay, now let's see the initiation phase, yeah, initiation phase. Yeah, this is the phase where the apoptosis process initiates, yeah. The pre-preparations are done, the activation of enzymes, factors are done, yeah. Okay, and in this phase also it is of two pathways, the very important pathway here, guys, the intrinsic pathway and extrinsic pathway. Intrinsic pathway is also called mitochondrial mediated pathway, yeah, because here in this process mitochondria are involved, yeah, major role of mitochondria are there. And in the extrinsic pathway many receptors functions here, yeah, and it is called death domain pathway. Okay guys, and we are going to discuss these both pathways one by one. Okay. Okay. Okay, now let's start with the intrinsic pathway. Guys, the term itself says intrinsic means something from interior side, yeah, from inside of the cell. Yeah. So guys, whenever there is any stress or any DNA damage or any abnormality inside the cell, okay, then it creates a stimulus or signals and it is received by the cell on its own and decides for the apoptosis process. Okay, and we are going to discuss in detail as well. Okay, so now let's start. Here you can see the intrinsic pathway is the major mechanism of apoptosis in mammalian cells, and its role in a variety of physiological and pathological processes as well is well established. Yeah, and this pathway of apoptosis is the result of increased mitochondrial permeability. Very important term here, guys. Mitochondrial permeability means here the pores are formed in the mitochondrial membrane. Yeah, in this process, in the middle of the process, the mitochondrial pores are formed, and out of which cytochrome C, the very major protein here, the cytochrome C releases outside of the mitochondria into the cytoplasm, and then the further processes takes place. Okay. Okay. Mitochondrial permeability and pro-apoptotic molecules that are death inducers in the cytoplasm. Guys, there are two specific molecules. They are, one is a pro-apoptotic molecules, another is anti-apoptotic molecules. Okay, this is the basic term here. And pro-apoptotic molecules are those molecules that are death inducers. Yeah, they supports apoptosis. Okay, and another one is anti-apoptotic molecules. The term itself says anti means it resists the apoptosis, means they are apoptosis inhibitors. Okay. Okay, so now here you can see cytochrome C is an important mitochondrial protein necessary for the essential life, but when released into the cytoplasm it initiates apoptosis. Yes, guys, it's a very important term here. Usually the function of cytochrome C is to bind the mitochondria and support the electron transport chain, yeah, which is a very important pathway, cellular respiration in our body. But because of certain regions, whenever cytochrome C releases outside the mitochondria into the cytoplasm, then it acts as a toxin for the cell. Yeah, it should not happen, and if it is happening, if it is releasing inside into the cytosol, then it is self understood that the cell must be dying. The cell is preparing for the apoptosis process. Okay guys, yeah, this is the basic idea, the basic concept. Okay, now let's move on. Okay, the release of these mitochondrial proteins, that is cytochrome C, is controlled by balance between pro and anti apoptotic factors of the BCL family of proteins. Okay, so guys, we have discussed about the pro and anti apoptotic factors here. Yeah, and these both are regulated by the BCL family of proteins. Yeah, they are the regulators of apoptosis actually. Yeah, very important proteins there, guys. Okay. Okay, there are more than 20 BCL proteins and their functions to regulate apoptosis. Yeah, very important. The anti-apoptotic proteins, they are BCL2, BCL-XL, MCL1, normally resides in the cytoplasm and mitochondrial membrane. Okay guys? Yeah, and these anti-apoptotic proteins that control mitochondrial permeability and prevent leakage of mitochondrial proteins that have ability to trigger the cell death. Yeah, guys, we have already discussed about the anti-apoptotic molecules. Means they are the molecules that inhibit the apoptosis process. And how they inhibit? They inhibit by preventing the leakage of mitochondrial proteins that are cytochrome C from the mitochondria. Yeah, actually, which triggers the cell death. Yeah, okay. When a cell is deprived of critical survival signals, or there is damage in DNA or misfolded proteins, then the sensors of damage or stress are activated. Okay, guys, actually what it is saying is, whenever there is any stress going on inside the cell, yeah, or there is a DNA damage, or there is a oxygen deprivation or hypoxia, yeah, then there are many sensors inside the cytosol, yeah, they start sensing the stress, yeah, and they start activating the other proteins, yeah, for the cell death. Yeah, this is the function. Okay, these sensors of damage are also proteins that belongs to the BCL family of proteins. Yeah, guys, very important family it is. Okay, the activated sensor proteins used in apoptosis include beam, bead, bad, and another one is back and backs. Okay, and these sensor proteins in turn activate the pro-apoptotic factors. They are back and backs. Pro-apoptotic factor means they are death inducers, supporters of apoptosis process. Yeah, remember? Yeah, guys, means these sensor proteins will activate back and backs, and the function of backs and back is in proliferation of mitochondrial membrane. Yeah, means they will create pores in the mitochondrial membrane, and out of which cytochrome C will escape. Yeah, cytochrome C will come out into the cytosol, and further process of apoptosis will take place. Okay, guys, this is the idea, this is the concept. Okay, now let's move on. Okay, so here you can compare now when and why it happens. Here, okay, here you can see this is the viable cell and this is the apoptotic cell. Okay, here you can see there is no leakage of cytochrome C. Yeah, in the normal cell, there is no leakage of cytochrome C. Yeah, means there is no any proliferation in the mitochondria. Okay, and which is also regulated by this BCL2 protein. Okay, guys, means here production of anti-apoptotic proteins takes place, means it is resisting, inhibiting the normal cell to go for a apoptosis process. Yeah, guys, yeah, and in the apoptosis process, here you can see in the apoptosis process, whenever there is any DNA damage or any sorts of stress going inside the cell, then there is activation of sensors, as we have already discussed. There is activation of sensors like BS3 only proteins. Yeah, and then they will create they will activate actually backs and back. Yeah, and this will make pore. Here you can see this is the pore in the in the outer membrane of mitochondria, and out of which there will be leakage of cytochrome C and other proteins. Activation of caspases takes place and apoptosis will occur. This is the idea, guys. Okay, okay, now let's move on. Okay, so the activation of pro-apoptotic molecules coupled with the loss of anti-apoptotic proteins causes the release of cytochrome C. Guys, actually whenever there is high concentration of pro-apoptotic molecules than anti-apoptotic molecules, then only the cytochrome C will release. Yeah, concentration gradient, it is actually a talk of concentration gradient here. Yeah, whenever there is a high concentration of pro-apoptotic molecules than anti-apoptotic molecules, then the release of cytochrome C will take place, means the cell will go for a apoptosis process. Okay, now once cytochrome C is released into the cytoplasm, it binds with the APAF, another important factor protein here it is, guys, that is apoptotic protease activating factor, yeah, and that will further form apoptosome. Apoptosome is a complex, yeah, it is a very important complex for activation of further caspases, yeah. And apoptosome is a complex formed by the accumulation of many proteins and molecules like cytochrome C, FF1, yeah. Okay, now at last what it does is apoptosome formed causes the activation of caspase 9. Caspase 9, okay, and this is a very important caspase here in the intrinsic pathway, yeah. This caspase 9 will further activate caspase 3, okay, and caspase 3 will finally lead to the apoptosis process. This is the idea, this is the concept here, guys, okay. So this is the major caspase of intrinsic pathway, that is caspase 9, and in extrinsic pathway it will be caspase 8, okay guys. Okay, now let's discuss about the extrinsic pathway also. Okay, now extrinsic pathway. So guys, the major caspase we have obtained from the intrinsic pathway is caspase 9, okay, and now the major pathway, the major caspase we will obtain from this extrinsic pathway will be caspase 8, clear? Okay, okay guys, so extrinsic pathway, guys, the term itself says extrinsic, means something from exterior, yeah, something from outside of the cell, means here the signal for the cell death comes from outside of the cell, yeah, given by other cell to the apoptotic cell, yeah, and there are many receptors on the cell membrane, yeah, and that receptor which are called death receptor, actually they receive the receptors, and according to that downstream processes takes place and apoptosis process begins, okay. So this is the idea, this is the concept, okay. This pathway is initiated by the engagement of plasma membrane death receptors on a variety of cells, okay guys. Okay, with the extrinsic pathway, the cross-linking between the death receptors and their ligand proteins causes the activation of caspase 8, yeah. Caspase 8 is another important caspase, yeah, we have discussed, and this caspase 8 will further go for the activation of caspase 3, and this caspase 3 will lead for the apoptosis process, very important guys, yeah. Okay, now guys, these are the ligands and they are the receptors. Ligands are the molecules, yeah, signal molecules actually, yeah, and for example FASL, TNF, they are the death ligands, and these are their corresponding receptors, yeah. Okay, okay, now let's discuss this big picture, yeah, of extrinsic pathway for more clearance. Here you can see this is the one receptor, this is one receptor, okay, and whenever any death ligands, suppose this is FASL ligand, will bind to this receptor, then what actually happens is all the similar type of receptors on the cellular membrane, yeah, all the similar type of receptor on the cellular membrane gets accumulated with each other, yeah, they forms a close proximity with each other, yeah, and forms a complex. Here you can see there was one receptor, when it bounded with one ligand, then all the similar type of receptor bound together and formed a complex which is called disc, which is called disc. Disc is nothing but a death induced signaling complex. Okay, guys, death induced signaling complex. Okay, after to that, what happens is it activates the death domain, which is inside the intracellular membrane. Yeah, and this death domain, which is also called FADD, fast associated death domain, okay, will further activate procaspase 8 by the autocatalytic caspase activation and activate the caspase 8. Here you can see procaspase 8 converts into caspase 8. Okay, and this caspase 8 will go in the executioner phase and form necessary caspase 3, and then the apoptosis process will take place. This is the idea, this is the concept. Okay, guys. Okay, now let's move on. Okay, so now we have finished the initiation phase of apoptotic pathway. Yeah, we have discussed the intrinsic and extrinsic pathway. From the intrinsic pathway, we have obtained caspase 9 as a major protein, and from the extrinsic pathway, we have obtained the major protein. The caspase is caspase 8. Okay, guys. Now we have to go in the execution phase. Okay, here the two initiating pathways converge to a cascade of caspase activation which mediates the final phase. Okay, the two initiation caspases are caspase 9 and caspase 8. Yeah, guys, from intrinsic pathway caspase 9, from extrinsic pathway caspase 8. Okay, and now these both caspases will finally converge to a common cascade of caspase, that is caspase 3. Okay, and this caspase 3 will execute the further apoptotic pathway, further apoptotic process. Okay, okay, the activation of executioner caspase, that is caspase 3 and 6. Okay, the executioner caspases once activated eventually leads to degradation of cellular cytoskeleton, nuclear matrix, and promote nuclear fragmentation, ultimately leading to cell death. Okay, so guys, this is a very major term we need to understand here. Here actually what happens is, whenever there is a release of or activation of caspase 3, then it activates the nucleases and proteases enzyme. Yeah, actually it clips the substance that inactivate that were inactivating the nucleases and proteases. Okay, and once they are activated, the nucleases breaks down the nuclear fragmentation, that is they breaks down the DNA, RNA, and other nuclear proteins. Okay, and proteins will disintegrate or will break the necessary proteins like as actin, microtubulins. Yeah, means finally they will break the cytoskeleton of the cell. Now once the nucleus and cytoskeleton is broken or cleaved, means the cell starts disintegrating. Yeah, and once they are disintegrated, the further phagocytic cells will come, they will engulf the cellular debris, and finally the cell will die, means the process of cell death completes. This is a very important concept and idea, guys. Okay, okay, now let's move on. Okay, now let's see this big picture, the comparative picture of extrinsic pathway and intrinsic pathway, and now let's recollect all the ideas of intrinsic and extrinsic pathway. Okay, guys. Okay, now let's start with this extrinsic pathway, then we'll talk of intrinsic pathway. Okay, in this extrinsic pathway we can see there is a death ligand coming from outside of the cell. Yeah, and there are receptors which are forming a complex which is called disc. What was the disc, guys? Death induced signaling complex. Okay, okay, now this will activate the death domain inside the intracellular membrane, that is FADD. Yeah, Now this FADD will activate the caspase 8 and 10, means procaspase 8 and 10 will convert into caspase 8 and 10. Okay, now this is the major protein here. Yeah, this is the major protein here, and after to that, here you can see there are two pathways. Yeah, this is the direct pathway by which the caspase 8 and 10 will get converted into caspase 3 and 7. Yeah, and finally it will go for a apoptosis pathway. This is one idea, one path. And another path is here you can see from the bead, from the activation of bead, T bead, by the lots of complex processes, it will again form caspase 3 and 7, and again it will go for a apoptosis pathway. So what it is trying to say is here caspase 8 and 10 is the intermediate caspase protein between both these extrinsic and intrinsic pathway, isn't it guys? Because it is cross talking between both of these pathway, extrinsic pathway and intrinsic pathway. Yeah, very important concept here it is guys. Okay, so now let's discuss it more. Okay, so here let's talk about the intrinsic pathway here now. So guys, whenever there is any intrinsic lethal stimuli, means any sort of DNA damage, ER stress, hypoxia, means oxygen deprivation, lack of oxygen, or any type of metabolic stress going inside the cell, okay, then it gets sensed by the many sensor proteins, and they are usually BS3 only proteins. They will sense it and then will further activate the other proteins, like it will activate first P53 protein, very major protein we have discussed in our first part of the video. Yeah, P53 protein, because they only decide for the apoptosis process. Yeah, they will decide whether the apoptosis will take place or not. Okay, so after it gets sensed, it activates the backs and back, very major protein here, and this backs and back will further proliferate, will create pores inside the mitochondrial membrane. Yeah, here, and out of these pores that are called MOMP, mitochondrial outer membrane permeability, okay, MOMP, out of this MOMP cytochrome C will release here, and this cytochrome C will bind with the APF1 protein and forms a complex called apoptosome. Very important complex it is guys. Yeah, and this apoptosome will further activate caspase 9, and this caspase 9 will activate caspase 3 and 7. Yeah, and finally the cell will start disintegrating, the nucleases and proteases will start activating. Yeah, means the substance that were inhibiting the nucleases or inactivating the nucleases or proteases gets cleaved, gets cut out, and the nucleases and proteases are activated, and they will start disintegrating the nucleus and cytoskeleton of the cell, and finally they will disintegrate. There will be the formation of many blips, and then finally other phagocytic cell will come and they will engulf the cellular debris, and the cell dies and the apoptosis process completes. Okay guys, so this was the pathway of intrinsic pathway, and this is the pathway of extrinsic pathway. Okay, now let's discuss about this pathway from the caspase 8, the intermediate pathway, crosstalk. Okay, so here whenever there is activation of caspase 8 and 10, then this will activate the bead, a protein, and then again this bead will be activated as a truncated bead, T bead, and this T bead will again activate the backs and back, and again the whole process will continue as the intrinsic pathway. Yeah, and the cell finally dies. This is the idea, this is the concept here guys. Okay guys, so this was the big picture of comparison between these two pathways, extrinsic and intrinsic pathways. Okay guys, okay, now let's move on. Okay, so for more clearance, let's see the concept once again. Yeah, let's discuss in this big picture. Okay, so let's discuss about the intrinsic and extrinsic pathway one by one. Here you can see Why intrinsic pathway takes place? Because of many cell injuries, any stresses, DNA damage. Okay, it gets first sensed by the BCL2 family sensors, then BCL2 family factors back and backs activated, then proliferation is done in the mitochondria, and then cytochrome C will release, then apoptosis will form, then caspase 3 is activated, and further apoptosis process takes place. Okay, but here in the receptor extrinsic pathway, receptor ligand interaction takes place, adapters will come, finally endonuclease activation, breakdown of cytoskeleton will take place, and the nuclear fragmentation and cytoskeleton breakage will occur, and cytoplasmic blebs are formed, apoptotic bodies, and then finally it is engulfed by the phagocytic cell, and the apoptosis pathway completes. So guys, this is the full concept on apoptosis pathway here. So go through the video once again. We will have a clear cut concept on apoptosis till now. Yeah, okay guys, so this was all about. Yeah, so if you found the video effective, please hit the like button, do share with your colleagues, and yeah, subscribe the channel. Okay, okay guys, so this was all about. Take care, bye bye.